Hi everybody. So today we are discussing on another new topic, that is monoclonal antibodies and their applications. Let us see what is a monoclonal antibody. So generally the body's immune systems attack the foreign substances by making a huge number of antibodies in our body. So, an antibody is a protein that sticks to the specific protein called antigens. So, antibody, they are produced by the body and it works by sticking with the specific antigens. So, antibodies circulate throughout the body until they attach with an antigen. So once they attach with the antigen, they can force other parts of the immune system to destroy the cells containing the antigen. So generally, the antibodies that are produced by the human body or the immune system, which attacks the antigen, and it also forces the immune system to destroy all the antigens in our body. So the designing of antibody, we can design a researcher, a scientist who works with the antibodies can design antibody that can specifically target to the particular cells, especially the diseased cells. So they can make many copies of such type of antibodies, and those we call it as the monoclonal antibodies. In other way, so monoclonal antibodies are the antibodies designed or developed by the humans in order to attack some specific kind of antigens. So such type of antibodies developed by the humans or the researchers, we call it as the monoclonal antibodies. So these antibodies are used to treat many diseases like cancer. The monoclonal antibody, to treat with this monoclonal antibody, it should be able to identify the target first. Target in the sense to which antigen it has to bind. And binding of such type of targets for many of the diseases is not an easy task. What the monoclonal antibodies are made of? So the monoclonal antibodies are made of 4 different ways, which includes, and they are known as the murine, the chimeric, the humanized, and the human. So let us see one by one what are those. Murine means these are made from mouse proteins, and the name of the treatment ends in OMAB, that is OMAB, that is at the end of the monoclonal antibody it will be mentioned as OMAB, which means they are the antibody developed from the mouse proteins. They are known as the murines. Next one is the chimeric, where that include a combination of both mouse as well as human. So the protein combination, a part of antibody will be the mouse part and another part will be the human part. And the nomenclature, it ends up with the word XIMAB, XIMAB. The third one is the humanized. So these are made from small parts of mouse proteins attached to the human proteins, and it is named as ZUMAB, ZUMAB. The last one is the humans, where they are fully human proteins and named as UMAB, that is UMAB. Now, what are the different types of monoclonal antibodies used? They are naked monoclonal antibody, conjugated monoclonal antibody, and Bispecific monoclonal antibodies. What are naked monoclonal antibodies? By the name itself, this represents that is no drug or no radioactive material attached to them. That is, they are purely certain monoclonal antibodies without any sort of drug molecules attached to it. So these are the common type of monoclonal antibodies used to treat the cancer. So most of the monoclonal antibodies attach to the antigens on the cancer cells, but some work by binding to the antigen or other. So the non-cancerous cells are even free floating proteins. The naked monoclonal antibodies works in different ways. Some of the MABs boost the immune response system against the cancer. So this is one way it attack. So an example of such type of antibodies available. The first one is the Campath, that is a marketed name, which is used to treat the patients with the chronic lymphocytic leukemia, and this functions by binding to the CD5 antigen found on the lymphocytes. So once this camphath attached to the CD5 cells, the antibody attracts the immune cells to destroy these antigens. So as this MAB binds with the CD5 antigen, it promote or it force the human immune system to cells these antigens. Some other naked MABs boost the immune response by targeting the immune system checkpoints. So this antibodies directly force or increases the response of the immune system. Some other works by attaching to the blocking antigens on the cancer cells that helps the cancer cell grow or spread. So as we discussed, the naked MABs works by this different method, and the third type of method wherein which this MAB binds with the antigens on the cancer cells. This antigen may be the cause for fast spreading or low or fast growth of the tumor cells. Once this antibody binds with such type of antigens, it reduces the growth of the tumor cells. An example of such type of antibody is Herceptin. So Herceptin is an antibody against HER2 protein. The breast and the stomach cancer cells have the large amount of this HER2 proteins. When HER2 is activated, it helps the cells to grow. And this Herceptin binds to the proteins and stop them from becoming inactive. Okay, so this way the Herceptin monoclonal antibody functions in order to treat the breast and the stomach cancer. It basically binds with the HER2 proteins. So when it is HER2 proteins are activated, this inhibit the growth of the cells, cancerous cells. The another class of antibodies are the conjugated monoclonal antibodies. Conjugated monoclonal antibodies are combined with chemotherapy drug or a radioactive particle. These monoclonal antibodies are used as the homing devices to take one of these substances directly to the cancer cells, and it circulates throughout the body until it finds a hook into target antigen. So, conjugated monoclonal antibodies are conjugated with either the drug or with any of the radioactive particles. Once it binds with the corresponding antigen, it releases the drug molecule. Until it binds with the antigen, it circulates within the systemic circulation without releasing the drug molecules. So these antibodies are activated only once it is conjugated with the only once it is bound with the antigens and it releases the conjugated drug molecules. It then delivers the toxic substance where it is needed the most. The toxic substance represent here it is the drugs or the chemotherapeutic drugs. So this method lessens the damage to the normal cells in other parts of the body because this exactly binds with the antigens. So these conjugated monoclonal antibodies are also sometimes referred to as stacked, labeled or loaded antibodies. Since they have drug molecules with that, they are called as either a stacked, MAB, labeled or loaded antibodies. Next one is the radio labeled antibodies. So radio labeled antibodies were in which the antibodies are conjugated with the radioactive particles. Example is Zivalin. So this antibody works against CD20 antigen which found on the lymphocytes called B cells. This antibody delivers the radioactive material directly into the cancer cells. It is made of monoclonal antibody drug and a radioactive substances. That is, the drug will be rituximab and the radioactive substance will be yttrium-90. So treatment with such type of radioactive substances we call as radioimmunotherapy or RIT, where the radioactive substance is conjugated with the monoclonal antibody. The drug and the radiation are delivered directly to the target cells called target cells. So here the target cells or the causative cells receives both the effect of drug as well as the effect of a radiation. The next one is chemo-labeled antibodies. These monoclonal antibodies have powerful chemotherapeutic drug attached to them. Example is brodiximab vedotin. So it's an antibody that targets the CD30 antigens attached to the chemo drug called MMAE. So this antibody that targets the HER2 protein attached to the chemo drug called DM1. So how this chemo labeled antibodies works? The main speciality of the chemo labeled antibodies is they are having strong or specific chemotherapeutic drugs. Once this antibody binds with the antigens, especially CD30 antigens, it releases the chemotherapeutic drug molecule which gives the complete effect of chemotherapy without harming the normal cells. Next one is the bispecific monoclonal antibodies. So these are the drugs made up of two different monoclonal antibodies, meaning they can attach to two different proteins at the same time. The example is Blincyto. It is used to treat the leukemia. So as mentioned, the bispecific monoclonal antibodies they can at a time they can attach with the multiple antigens. The all that type of antibodies what we discussed earlier can bind only with a single antigen once it is administered. But these bispecific monoclonal antibodies, they're having two sites to two antigens which can bind. So one part of this drug attached to the CD19 protein, which is found on some leukemia and lymphoma cells. It other parts bind to CD3, a protein found in the immune cells called T cells. So by binding to both of these proteins, these drugs bring the cancer cell and immune cell together, which is thought to cause the immune system attack the cancer cells. So this way, the bispecific monoclonal antibody, which can bind to the CD19 cells as well as the CD3 cells. So this way, just by administering this antibody, it can attack both the cells and can protect the normal cells and decreases the progression or the growth of the cancerous cells. So these are the various types of monoclonal antibodies. So that's all for this session. Next session we will continue with the various applications of monoclonal antibodies. Thank you.